This chapter will focus on the defenses of the host. The immune system is a complex organization of non-specific functions that we're born with, as well as the adaptive system, which is acquired and has specific functions. We will discuss them as our first, second, and third lines of defense. Our first line of defense includes anything that can prevent entry of a pathogen and limit their invasion of tissues. As good as it is, it does not recognize substances and has a generic mode of action. The second line of defense includes cells as well as fluids. This system is activated when a pathogen breaches that first line of defense. It also includes both inflammation as well as phagocytosis. The third line of defense is acquired upon exposure to a pathogen, and it provides a specific response along with long-term protection. The innate system is something we are born with and is non-specific. It includes the first and second line of defense. The first line is comprised of barriers to prevent entry. It includes physical barriers, normal flora, and chemical barriers. The second line of defense includes cells, phagocytosis, inflammation, fever, and proteins. The third line of defense is acquired and is specific. It includes cells that take part in the second line of defense, as well as B cells and T cells. The systems tend to overlap and sometimes are similar in terms of their effects. The cumulative effect of these three systems makes it very difficult for a pathogen to invade. Immunology focuses on studying the second and third lines of defense, how the host responds to pathogens, as well as allergies and cancer. If an immune system is functioning properly, it is constantly on patrol, searching for microbes. It also is responsible for recognizing anything foreign and for destroying it. White blood cells are on the hunt for anything that could possibly be a pathogen. They also have the ability to recognize the host's own cells and differentiate them from non-self cells. This process is a major part of the immune system, and the loss of the ability to recognize self from non-self can result in autoimmune diseases where healthy cells are attacked. So how do these cells recognize self from non-self cells? They look for markers, also known as antigens, that are made of protein and/or sugars. These markers are found on the surfaces of cells, and our immune system uses them to determine if it's a possible threat. And if it is, they attack via phagocytosis. PAMPs, or pathogen-associated molecular patterns, are markers found on the surfaces of microbes. Host cells have pattern recognition receptors that recognize PAMPs and are part of the second line of defense. Anything that's non-self and not a threat are not targeted by the immune system. When you think of a system like the cardiovascular system, you think of things like the heart and blood vessels. The immune system is different in that it's made up of cells and fluids that are everywhere. This provides optimum protection by being able to be on patrol everywhere. In order to have an effective response, there must be efficient communication throughout the body. At the microscopic level is the mononuclear phagocyte system. It is a cluster of cells that have direct contact with this system, along with extracellular fluid. Blood capillaries and lymphatic capillaries invade these tissues, and it allows for cells and chemicals of this system to hitch a ride in blood and lymphatics. The lymphatic system is made up of vessels, cells, and specialized organs. It's responsible for transporting lymph through vessels and lymph nodes, and then ultimately dump the lymph into the circulatory system. Let's look at the circulatory and lymphatic systems. White blood cells circulating in the cardiovascular system screen for pathogens. The lymphatic system is comprised of vessels and organs like the lymph nodes and spleen. The lymphatic system runs parallel to the circulatory system, but only travels in one direction. The purpose of the lymphatic system is to return fluids to the circulatory system and serve as a drain-off system during inflammation. It also provides surveillance locations for immune system cells. The lymphatic fluid is very similar to plasma and is formed when blood components travel from the blood vessels through the extracellular space and then through lymphatic capillaries. It also transports white blood cells, fats, waste, as well as infectious agents. This explains how cancer can move from one location to another. The lymphatic vessels are very similar to veins and accompany the blood capillaries. They make their way into a variety of spaces in the body, with a few exceptions, and their walls can easily be breached by extracellular fluid. We have an abundant amount in our hands and feet, as well as around the areola. The lymph travels to the heart via contractions of skeletal muscles. The primary lymphatic organs are areas in which immune cells are born and mature. They include the red bone marrow and thymus. The secondary lymphatic organs are areas where immune system cells are activated and where they reside and function. They include the lymph nodes, spleen, and other lymphoid tissue. The red bone marrow intersects the circulatory, skeletal, and lymphatic system. It's found within the matrix of long bones and is where blood cells are produced. After B cells mature, they leave the bone marrow to the secondary lymphatic organs. The thymus is a location in which the T cells mature. It originates in the lower neck region during embryonic development. Once the T cells develop specificity and mature, they settle in the lymph nodes and spleen. The lymph nodes can be found in the axillary region, the groin, and neck. When they enlarge, it's indicative of an infection. The spleen can be found in the upper left region of the abdominal cavity and is responsible for filtration of blood and for the removal of old red blood cells. It can also filter pathogens using macrophages, which are phagocytic. If an adult lacks a spleen, they live a normal life. However, if a child has their spleen removed, they experience a decreased functioning of their immune system. Skin-associated lymphoid tissue and mucosa-associated lymphoid tissues contain lymphocytes that can provide a defense against microbes from food and air. Tonsils also contain lymphocytes, and the breast, in some situations, have antibodies. The gut-associated lymphoid tissue can be found in the appendix, lacteals in the intestinal villus, and Peyer's patches in the ileum of the small intestine. Whole blood is made up of blood cells and plasma. Serum is practically the same as plasma, but is clear and comes from clotted blood. Hematopoiesis describes the production of blood cells. Stem cells give rise to new blood cells and can be found in the bone marrow. They proliferate and then differentiate or transform into the specialized mature cell. The stem cells give rise to red blood cells, platelets, as well as white blood cells. The white blood cells, or leukocytes, are characterized by their staining patterns and are important in both nonspecific and specific immunity. All cell types in the blood originate from the hematopoietic stem cell. The leukocytes, which are the white blood cells, are characterized as agranulocytes or granulocytes. The agranulocytes are those that don't contain granules in their cytoplasm. They include the monocytes, which transform into phagocytic macrophages and dendritic cells. The agranulocytes also are the lymphocytes, which include B cells, T cells, NK cells, and gamma delta cells. The granulocytes which have granules in their cytoplasm, are your first responder phagocytic neutrophils, the basophils, eosinophils, and mast cells. The hematopoietic stem cell also gives rise to red blood cells and platelets. Cytokines are molecules that have a role in cell development, inflammation, and immunity. Immune system cells are responsible for their production, and the effects may be localized or systemic. They may also provide short-term or long-term actions, may be specific or non-specific, and could have protective properties or could cause damage to the host. Some cytokines, like interleukin-1, encourage inflammation and immune responses. Interleukin-10 is an anti-inflammatory cytokine that actually discourages the response from the immune system. Histamine acts as a vasodilator that increases the diameter and permeability of blood vessels. Other cytokines, like interleukin-7, can stimulate production of lymphocytes. Tissues of the body contain connective tissue fibers or reticulum. They connect cells and meshes with the connective tissue. Here we find phagocytic cells. You can see that the degree of shading shows variations in the concentration of phagocytes. This mononuclear phagocyte system can be found in the thymus, lymph nodes, and tonsils, just to name a few. This system consists of phagocytic macrophages just waiting to find a pathogen and attack. The first line of defense is something we're born with and is non-specific. It includes both physical as well as chemical barriers that prevent entry of pathogens. The image shows these primary physical and chemical barriers. The blue arrows show the direction of fluid flow. Let's look at these built-in defenses of our skin. The outer layer, the stratum corneum, is pretty impenetrable and waterproof. We are also constantly shedding skin, which eliminates microbes. Shedding of our hair shaft, as well as sweating, also helps to remove microbes. Our mucous membranes also provide a defense system. The mucus prevents entry of pathogens, as well as attachment. When we blink and produce tears, we flush microbes off the surface of our eyes. Saliva helps to transport pathogens to our stomach, where the acidity helps in their destruction. Vomiting and defecation also help to eliminate substances as well as microbes. Within our respiratory tract, large substances are trapped by nasal hair, and the mucus and fluids flush the pathogen. The ciliated cells transport trapped particles to the pharynx. Sneezing and coughing also helps in the elimination of irritants and pathogens. Here we can see a trapped bacterium in the epithelial lining, and the sweeping motion of the cilia transport it to the pharynx. Urine trickling and the emptying of the bladder help to eliminate pathogens. The vaginal secretions also help to cleanse the lower reproductive tract in women. Our normal flora also contributes to a structural barrier. It can block the entry of pathogens. The normal microbiota competes with pathogens for nutrients and space. Overuse of antibiotics has contributed to diseases like Crohn's disease and ulcerative colitis. There are also non-specific chemical defenses. The sebaceous glands secrete substances that inhibit the growth of microbes. The eyelids secrete substances that lubricate the conjunctiva and are antimicrobial by nature. The tears and saliva have a substance called lysozyme that break down the cell walls of bacteria. Sweat, acidic pH, and fatty acids are also antimicrobial. The stomach and intestines also provide chemical defenses along with semen and the pH of the vagina. So what happens if the barrier is lost? If a patient has severe burns with the barriers destroyed, they become more vulnerable to infections. Blockage in glands, as well as the tear ducts, intestine, and urinary tract, also lead to an increased risk of infection. This first line of defense is great, but by itself, it does not provide sufficient protection. The second line of defense is also necessary. The second line of defense is nonspecific as well and involves cells, the inflammatory response, fever, and proteins. Phagocytosis involves immune system cells searching, identifying, and ingesting and eliminating pathogens. The phagocytes are also responsible for extracting antigens. The phagocytes include neutrophils, monocytes, and macrophages. Neutrophils are phagocytic leukocytes that are nonspecific. They're the first responders in the inflammatory response, and a high count indicates a bacterial infection. Neutrophils are also a primary component of pus. Monocytes are another type of leukocytes that are phagocytic. They differentiate into macrophages once they make their way into the tissues. Specialized macrophages called histocytes live and remain in certain tissues their whole life. They include alveolar macrophages in the lungs, Kupffer cells in the liver, dendritic cells in the skin, as well as others in various locations. Some don't plant roots, but are nomadic and travel around. Here we see the stationary alveolar macrophages, Kupffer cells, and Langerhans cells, Langerhans dendritic cells. Phagocytes eat cells. The process of phagocytosis includes 5 steps, which we will talk about in detail on the next slide. The first step in phagocytosis is chemotaxis. During this stage, the phagocyte moves to the inflammatory site by following a gradient of products released. The second step is adhesion, where phagocytes use the pattern recognition receptors to identify PAMPs on the surface of the pathogen. They then adhere to one another. The next stage involves engulfment and phagosome formation. After attachment to the pathogen, pseudopods bring the pathogen into a vacuole called a phagosome. The next step involves the fusion of the phagosome with a lysosome. The lysosome releases antimicrobial chemicals into the phagolysosome. The chemicals induce a respiratory burst, which destroys and kills the bacterial cells. The waste are then released via exocytosis. PAMPs are recognized by phagocytes and other immune system cells. We as mammals do not have them, so they serve as a red flag to our immune system. Bacterial PAMPs include the peptidoglycan in the cell wall and the lipopolysaccharide. Viral PAMPs are the double-stranded RNA. The host has pattern recognition receptors which can be found on immune system cells. They recognize PAMPs and bind to them. Inflammasomes are in the cytoplasm of phagocytes and recognize PAMPs after the pathogen has been phagocytosed. It then leads to inflammation. Several signs and symptoms are indicative of inflammation. Rubor is redness and is caused by increased circulation and diameter of blood vessels. Calor is warmth, which is due to increased blood flow. Tumor is swelling, and allure is pain, due to the excess fluid pushing on nerve endings. Occasionally, loss of function is seen. Some diseases like cardiovascular disease are caused by chronic inflammation. The inflammatory response can be either localized or systemic. Several factors can trigger inflammation. It may be trauma, tissue injury, or immune reactions. The main role of inflammation is to attract immune system cells to the site of injury. It's also involved in tissue repair and removal of harmful substances, along with destruction of microbes. It's a major defense reaction and does have the ability to damage tissues as well as cause disease. The process leading to inflammation can last minutes, hours, or even days. Let's discuss the inflammatory response. The first step is injury and the immediate reaction. After an injury, cytokines are released. Vasoconstriction occurs to prevent blood loss while immune system cells are called in. The blood vessels then increase in diameter, then constrict and dilate again. It helps to flush the bacteria away and prevent blood from leaving and bring additional immune system components in. This leads to the redness and warmth. Capillaries also become more permeable and components leak out, forming exudates. The accumulation of fluid leads to swelling. This fluid contains immune system components. Pus forms, and in some cases, fibrinogen is converted to fibrin. Neutrophils then crowd the site of injury. Resolution is the final step and can be complete, or scar tissue can form. Macrophages squeeze out of blood vessels by a process called diapedesis. We then see newly healed tissue. Diapedesis is again the process of white blood cells squeezing out of blood vessels. Chemotaxis is the use of a chemical stimulus, like a trail of breadcrumbs, that the white blood cells follow to a site of infection. Here we can see the white blood cells leaving the vessels during diapedesis. And then they follow a chemical gradient to the site of injury in chemotaxis. So why is edema or swelling beneficial? It helps to dilute toxic substances, and the fibrin clot can prevent the spread of microbes. Neutrophils help to phagocytize bacteria and waste. Pus also develops. Some bacteria like streptococci and staphylococci are pyogenic and enhance the formation of pus. Fever is an abnormally high body temperature and is a universal symptom of infection. Sometimes the fever has an unknown cause. The normal body temperature is 98.6 degrees Fahrenheit. A low-grade fever is 100 to 101 degrees Fahrenheit, and a high-grade fever is 104 to 106 degrees Fahrenheit. Pyrogens induce the hypothalamus to change to a higher setting. Exogenous pyrogens come from pathogens, blood, vaccines, or injectable solutions from outside the body. Endogenous pyrogens come from within, like from immune system cells during phagocytosis, as well as cytokines. We think of fevers as being bad, but they can be beneficial. They prevent replication of microbes that can't withstand high temperatures, and it takes iron away from microbes. It also speeds up the healing process by inducing hematopoiesis, phagocytosis, and immune reactions. Sometimes it's difficult to decide if a fever needs treatment. A low-grade fever in a healthy patient should be left alone. Some side effects seen by a fever include an increased heart rate and respiratory rate, as well as febrile seizures. Of course, if it's a high fever, prolonged, or seen in people with underlying conditions, it should be treated. Interferons are proteins produced by some white blood cells and tissue cells. They were once shown to only protect against viruses, but is involved in defenses that target microbes, as well as regulation of the immune system. Alpha and beta interferons are released by lymphocytes, fibroblasts, and macrophages, while gamma is released by T cells. So how do these interferons work? They exert their effects by binding to the surface of the cell and causing changes in genetic expression. All interferon types inhibit the expression of cancer genes. Alpha and beta interferons enhance the effects of phagocytes, and gamma regulates macrophages and lymphocytes. Anytime a virus or other pathogen binds to the host cell, the host then releases interferon. Interferon binding leads to the production of specific proteins that prevent viral replication by degrading RNA and preventing translation. It is nonspecific and can be used for treatment of different viral infections. On your left, you can see the infected cell. Once the virus enters the infected cell, it then produces interferon. In the meantime, new viruses are released and target a new host. A neighboring cell is then infected with the virus, but interferon has bound to its surface. It then begins producing antiviral proteins and prevents the virus from replicating. The complement proteins play a big role in assisting the immune response. The complement system is made up of over 30 different proteins that destroy pathogens. Each chemical activates the next, and so forth. There are 4 stages of the complement system. The first is the initiation stage, where C3, whether it be free or attached to a pathogen, is split into C3b and C3a. The next step is activation and the cascade, where the C3b protein breaks down C5 into C5a and C5b. Next is polymerization, where a membrane attack complex is formed. The membrane attack complex then forms pores or openings on the surface of the cell, which leads to lysis. There are different pathways that the complement system can be activated. The first route is the classical one, which involves antibodies binding to the surface of the pathogen. The alternative pathway is activated by the mere presence of an antigen and does not require an antibody. This generates a quicker response. In step 1, we can see C3 hydrolyzed into C3a and C3b. During activation, C3b splits C5 into C5a and C5b. In polymerization, C5b combines with other complement proteins to form the membrane attack complex, which then causes lysis of the cell. Antimicrobial peptides are comprised of up to 50 amino acids. They, like MAC, insert themselves into the cell membrane, forming an opening, and cause lysis of the cell. These products are being researched in how to utilize them in treating drug-resistant infections. Here we can see the antimicrobial peptides compromising the integrity of the cell membrane.